All right, I've been given the go-ahead by my AV team, so I think we're ready to start. Welcome, everybody, to the inaugural UCI School of Medicine Dean's Distinguished Lecture Series. I'm Dr. Michael Stamus, Dean of the School of Medicine here at UCI, and your host for tonight's event. I'm honored that you have all joined us this evening to hear from one of the most influential leaders in healthcare as she shares her insight, her experience, and vision for the future of regenerative medicine. Our speaker tonight is Dr. Maria Millan, as you well know. She's the president and CEO for the California Institute for Regenerative Medicine, commonly known as CIRM. She's a physician scientist who has devoted her entire career to treating and developing innovative solutions for children and adults with debilitating and life-threatening conditions. A medical doctor, Dr. Millan did her surgical training and her postdoctoral research in Boston at Harvard Medical School, Beth Israel Deaconess Medical Center. After a transplant surgery fellowship at Stanford University School of Medicine, she began her academic career with a busy pediatric and adult transplant surgery practice focused on technical advancements and optimization of patient outcomes. In parallel, she continued her bench research at Stanford and was soon promoted to associate professor and director of the pediatric organ transplant program. She has widely published in the areas of cell biology, immunology, and clinical organ transplantation. In 2006, with early signals from science about the potential of stem cells and regenerative medicine therapy, she ventured into the private sector to join Stem Cells Incorporated. And in December of 2012, she continued her focus on stem cell and regenerative medicine and joined CIRM, where she led the formation of the Alpha Stem Cell Clinics Network, a network of California medical centers that specialize in rigorous and high-quality clinical trials and top-tier medical care for patients participating in these trials. We are fortunate to have one of those clinics here on the UCI campus. In July of 2017, she took on the role as president and CEO of CIRM, and she was formally appointed by CIRM's board in September of that year. Under her leadership, CIRM has funded a total of 45 clinical trials as it continues to grow a robust portfolio of high-quality programs and accelerate stem cell treatments to patients with unmet medical needs. Please join me in welcoming our inaugural Dean's Distinguished Lecture Series speaker, Dr. Maria Millan. Thank you.
Thank you. Thank you very much. Thank you so much, Dr. Stamos, and thank you, Dr. Daniela Bota, who is our director for the UC Irvine Alpha Clinics, and she had kindly initiated the invitation. And it's wonderful to be here. I never thought I would be giving a lecture in a room with surfboards, beautiful ocean views behind me, and with a very engaged and robust audience this evening. So thank you for being here. I've had a wonderful day with the UCI faculty and continue to enjoy meeting all of you, the stakeholders, patients, advocates, and those interested in advancing science. So today the title of my talk is to give an update of how CIRM, through the vision of Californians and the creation of an agency that funds specifically stem cell and regenerative medicine research, has made progress in not only being part of what we now know is a transformative era in medicine, but has been a leader in this and is a recognized leader internationally. California is a leader in growing the field of stem cell and regenerative medicine. So because of your trust in the science, because of the bold vision of those in 2004, where we are today, and it's with great pleasure that I'm here to give you an update. I wanted to just kind of to use Oz Stewart, just to let you know we have board members from CIRM here. Dr. Oz Stewart, one of UCI's own, and Dr. John Thomas, the chair of our board, are both on our board. And later on this evening, after the lecture, we'll have the opportunity to have Q&A and discussions, and so it's wonderful to have them here. So Dr. Stewart likes to say unpack things. So I want to unpack the title a little bit and talk about, when we say accelerate, I want to make sure that we are very clear when we're talking about accelerating science to patients, It's not accelerating at the cost of quality or safety. It's maintaining the highest rigorous quality while still doing what we can to make sure we move the science because patients can't wait. There is an urgency, but patients can't afford to have bad things happen either. So accelerate, I want to make sure we qualify what we mean by accelerate. And then also I want to define what regenerative medicine is. Regenerative medicine is an approach where one is able to regenerate, restore, repair, or bring back to normal the physiology and the body's systems that have led to the disease. And so that's kind of it in general terms. And today, when we're talking about regenerative medicine, there are different platforms. I want to just say that I might be using things interchangeably, but it's generally using cells as a regenerative medicine therapeutic, or genes within the cells, either in the cells that one is transplanting or genes in our bodies to restore function and health. And then there's also tissue engineering as well. So that's regenerative medicine. Next slide. I think I can advance myself. All righty. So just by way of background, the California Institute for Regenerative Medicine was formed in 2004 with the passage of Proposition 71, which is a citizen-led bond measure that, through the approval by the citizens of California, had devoted $3 billion in bond funding to pursue something. And I want to just take a moment to put this in context. It was only in 1998 that the human stem cell, the human embryonic stem cell, but the stem cell was identified. The stem cell was shown that to be able to have an indefinite ability to propagate itself and to give rise to every cell and organ in our body. So you can imagine that that really opened up folks' imagination in terms of what can we do with this. We can study things that we couldn't study before. We could potentially even use derivatives of these stem cells to replace or repair disease. That was an idea, right? So that's 1998 as a science. There was an idea, and then there was this idea that, that can't happen unless people are working on this. It wasn't a field yet. So California, being California, said, well, we need to make that happen. And as you know, there were some ethical debates that were happening at the time. Things were happening. But meanwhile, and those things are important to happen, meanwhile, science needs to continue because we can have all these discussions, but unless it's studied, we don't know what the potential is. So in 2004, with the dedication of funds to be able to pursue the science, while all this was going on, we were able to then fund researchers to focus on this field. Otherwise, they would probably have to just work on something else. And able to fund all the different programs that I'm going to give you a little bit more detail on. So with that $3 billion, what's happened? Today, 1,000 programs have been funded. And I'll describe what those programs are. Throughout California at over 70 different institutions. It's the world's only dedicated funding source to fund stem cell regenerative medicine. And in the first 10 years of CIRM's existence, there were a total of 10 early clinical trials. Today, there are 56 clinical trials in this novel field that's being carried out, and I'll describe to you what indications, but it covers a broad array of different disease indications using the different platforms applying stem cells for the given indication, right? There are different types of products that have come out. And over 1,200 patients have been enrolled in these trials. The pictures you see here are some of the patients who've actually participated in the trial. Jake, on the far left, was in a spinal cord injury trial. At the time was being carried out by Asterias, which was a a company that took over from one of the early companies called Geron. And just by the way, now that technology has been taken up by another company that just went public called Lineage. So what I wanted to make a point of is the science, if it makes sense, will continue. It just takes different paths. And there is a devoted now ecosystem around this because of what we put in place with CIRM. Evie in the blue dress is now over 6 years out from being treated with a stem cell to correct her disorder and immune deficiency, and I'll describe that in a little bit. And that is this is a kind of a teaser, but that's a really significant project for a lot of reasons. Brandon in the striped shirt was born with what's called X-linked chronic granulomatous disease, where he has a defect in his neutrophils, the white blood cells that eat up bacteria and fungi, and, these things that we come across all the time. And because he had a defect in his neutrophils, isn't able to fight infections, had chronic abscesses, and formed antibiotic-resistant infections. It's a fatal disease. And he's out over 4 years, now having that been corrected with a trial, and I'll describe that. And then Rosie is one of the patients that was treated here at UC Irvine in a trial for retinitis pigmentosa, which is a blinding eye disease. So I mentioned that we've funded across different areas, the 1,000 projects, and I wanted to just describe what those projects are. So I love this feature. So infrastructure, which is building infrastructure, such as the Sue Bill Gross building here for stem cell research, was co-funded with CIRM to create a space where the research can be done. In addition, programmatic infrastructure, such as the Alpha Clinics, was funded under this program, as well as genomics and creation of what's the world's largest pluripotent stem cell bank, so different diseases can be studied using stem cell technology. The education program, I had the great fortune of having lunch with one of the first recipients of the training grant here, and he's now here as a faculty member because of that early work. And we also have programs for high school students and undergrads who at our Cal States, who now have the opportunity to work at places like UC Irvine, UCLA, Riverside, and have exposure in stem cell labs that they otherwise wouldn't have. So it provides a connectivity. The majority of the students who have gone through these programs have then gone on to either graduate work or get jobs in biotech because of these programs. Discovery, almost $1 billion. It's our largest investment. Good medicine starts with strong science. So that's always going to be the seed for what we give rise to down the line. And translation is when you take a discovery and you do the things that you need to do to show that this is really a candidate you're going to go after and that you can produce the cell or the gene product reproducibly so that it can be tested in patients. So that's a very unique role that CIRM plays, and it's known for this. It's something that the NIH is starting to try to do, but typically they fund mainly basic research, or on the other end, this in-between often gets left behind. And then we're having an increased investment in clinical trials. So $2.7 billion so far of the 1,000 awards has been expended, and almost $2 billion of that has been to develop therapeutics. If you look at what types of conditions or diseases or areas are being studied in these awards, you can see that it spans a huge gamut with about a third of it in different malignancies. Believe it or not, 30% in neurologic, and I want to get back to that. The total R&D spending in neurologic is significant. The number in clinical trials is not the same portion because it's still a field that needs to be, where more investment needs to go into to tackle some of the challenges And UC Irvine is a leader in some of these and actually has some programs already in the translational stage that we were talking about, as well as in the clinical. So, but you can see that it's a very heterogeneous and diverse portfolio that we have funded. And it's not, so this isn't predetermined, it's based on the science. Whatever is the strongest gets funded. And then organically, it goes into this distribution. I'm trying to advance, but it's okay. So all of those steps that go into taking the science, translating it, getting into clinical trials has led to 56 clinical trials tackling 35 different medical conditions in blinding eye disease, Lou Gehrig's disease. One of the trials that's run out of the Alpha Clinics network that Dr. Daniela Bota is leading is in ALS, or Lou Gehrig's disease, blood cancers, thalassemias, brain cancer, solid malignancies, a cure for HIV/AIDS, all sorts of genetic diseases, paralysis, stroke, diabetes. And mind you, this looks like a huge number of different indications. Just know that each of the different trials has had science behind it and has a technology platform that's appropriate for that indication. We're not saying there's one particular product and we're trying to throw it at everything. Not at all. One of the things I had alluded to in the beginning of the talk is that we are now in a very special time in medicine. We're at a very special time in medicine because the science in terms of understanding the genetics better, the stem cell biology in terms of understanding the biology of how cells work, what leads to them going into disease, what could repair them, has led to approaches that are more precise and then therefore also able to address previously insurmountable conditions more definitively. And I'll give you an example. The first child of this is called CAR-T. And it's not a new automobile, it's not a self-driving car. CAR-T is a chimeric antigen receptor T cell. T cells are immune cells that usually fight cancer. They're abnormal cells that float through our body all the time. But our immune system recognize it, and destroy them. When that doesn't work anymore, that's when cancers can grow. So that's called immune surveillance. And what the CAR-T does is take this natural biology that happens, but the T cells, the docking mechanism to recognize the tumors, are engineered in a way so now they're kind of a seek and destroy type approach. So now they can dock stronger and more specifically to the cancers, avoiding the need for chemotherapy, for instance, or making chemotherapy in lower doses more effective. So the first CAR-Ts were approved by the FDA just a little bit over 2 years ago, being developed by Big Pharma. In the meanwhile, CIRM has been funding programs in this arena and is now giving rise to the next generation CAR-Ts. And in fact, we have 5 programs in CAR-Ts that are targeting different antigens, the thing that the T cells dock to, as well as having more specialized T cells that stick around and are more persistent. So if there are little things if there's still cancer that persists or comes back, they can attack them. And those are just some of the programs that are part of the gene therapy portfolio that CIRM funded. We're also funding gene programs that target inherited diseases, HIV/AIDS, and I'll be talking about these programs a little bit more, but just if you remember that list, be going into that in a little bit more detail. And then you remember Evie, the girl in the blue dress? This is Evie just a couple of years ago. Why is this significant? Evie personifies this paradigm shift in medicine. So 6 years ago, it says 5 years ago, but 6 years ago, Evie was treated with her own blood stem cells. The blood stem cells out of her body were the ones that give rise to all the cells in the immune system and in the blood system, right? She was born with what's called bubble baby disease. And if you remember in the 70s, there was this movie with John Travolta where he was a boy in a plastic bubble and he had a girlfriend, but they couldn't really, interact in a normal way because he was in a bubble and she was outside. And do you remember that movie? Because anything, even a common cold, could lead to death, right? So I had a cold a week ago. I'm still suffering a little bit from it. But something like that cold could be fatal in these kids. And without treatment, it's fatal. Within 2 years, the mortality is really high. So Evie, through a trial that's being run out of UCLA, Dr. Don Cohn, is cured from this because her blood stem cells were taken out. The defect that led to her not being able to form these normal immune cells, called ADA, adenosine deaminase, it's a deficiency in an enzyme that leads to an inability for her immune system to form cells that attack viruses and things that come along. It was corrected, infused back into her through a regular IV. These stem cells are smart, so they home to the bone marrow, that's their home, and that's kind of command central where these blood stem cells give rise to all their progeny of the different blood cells, the immune cells that come out, and now they have they're now able to form normal immune cells. And so 6 years out, you took her blood sample, you'd see she has normal immune cells, they function, and she can go to school, run around without her parents hovering over her or trying to keep her isolated. So she just is one of 30 patients who've had the early experience. And the cool thing about this trial is it's what it's called a registration trial now. There are a lot of cool aspects of this. One of it is a registration trial is based on a few number of patients, whereas in the past clinical trials relied on hundreds and hundreds of patients, right? Well, in a rare disease like this, you can't, right? There are not enough patients. But it's not just a numbers issue here, but it's also because the biology of what you're correcting can be measured, whereas you couldn't do that with drugs. You're actually correcting the very thing that's leading to the disease. Because of that, you have a definitive marker or a definitive proof that it's working. And that's why the FDA can say, okay, it's really working. These cells, express the enzyme, they produce the cells, in addition to seeing what's clinically improved. Spectacular. What does this mean? A success for this shows that there's now a completely new approach that can just open things up for all genetic diseases, there's 6,000, 7,000 of them. So all these different rare diseases can now become, can be tackled by a platform that if you just make sure that you're looking at the right target, the genetic basis for it, you can use the same approach or similar approach. All righty. So Dr. Stamos had mentioned, and Dr. Bota are involved in the Alpha Clinics Network. It's a first of its kind clinical network where it focuses not just on a disease specific, although it addresses a whole host of different diseases, but it's a platform type network, meaning regenerative medicine. And it was a concept that was it's a very novel concept. The cool thing about it is it was a raging success. And because of the success of the UC Irvine, UCLA, San Diego, City of Hope initial experiment, we were able to expand it up to Northern California, to UCSF, Benioff, and UC Davis. And these clinical networks not only have the expertise in this, but they're creating a new field of nursing best practices, of specialized skills. And they're able to do things more efficiently. They're able to use shared IRBs. IRBs are the committees you need to go through to make sure that the trial is being looked at, monitored well. But there's only a certain number of experts, right? And so to kind of centralize that expertise speeds things up, because now an approval in one of these institutions could theoretically, the other institutions say, yeah, we approve it as well because we know it's being looked at the right way. And so it speeds it up. So when we were talking about acceleration, that's one of the advantages it has. But one of the trials that's being run out of the, I think almost 100 now that have been run out of the Alpha Clinics Network, are these first-in-human, really unique trials, including this trial. This is baby Ileana, born just a little bit over a year ago. Oh, spoiler alert. Ileana has what's called alpha thalassemia, major. alpha thalassemia major is a defect in formation of red blood cells that leads to death, often in utero, often to stillbirth, right? In this approach, Dr. Tippi McKenzie, who is a CIRM investigator, is a pediatric surgeon, because of her funding from CIRM, was able to take some time out of her practice to pursue an approach of transplanting Ileana with the mom's bone marrow, the blood stem cell from the mom, while she's still in the womb, to rescue her in terms of providing her the blood cells so she can get to term, but in addition, doing it in a way that increases the chances that it can engraft and become her own cells. Because in the womb, babies are more what's called immunologically tolerant, especially from the mom, which is a close but not an identical cell type. And so it's a convergence of the immunology of technical expertise, surgical expertise. Dr. Stamos and I are both surgeons, and Dr. Defoe in the audience. So we understand kind of the technical aspects of what it takes to make a transplant doable, or to make a complex colorectal surgery, being able to get that all in place. But in addition, working with the immunology. So this was actually a picture from the New York Times article, just highlighting what a spectacular advancement this is, and being able to put the biology of stem cells with surgical to tackle a fatal disease. So that trial's still ongoing, but it's a type of trial that requires really specialized resources, and that's something you have in the Alpha Clinics Network. Now, for somebody you'll recognize, Dr. Leslie Thompson, who's in the audience, with Francis Saldana and the other members of the HD community. I have to say that the formation of CIRM, as well as our continued work, is in partnership with our patient advocates. And they serve on our board. They are on our clinical advisory panels. They are in our review when we were looking at the science behind the projects. And so they're embedded in everything we do. And just during the reception, Dr. Thompson was there with the HD group working on this. So it's really remarkable. But in any case, Dr. Thompson's work is in Huntington's disease, which is a neurodegenerative disease that leads to mental deterioration early on, and then movement disorders, and it's just a devastating condition. It's inherited, and with each generation could actually afflict a little bit earlier. And we kind of know some of the biology behind this. It's not yet something that's directly correctable with a gene therapy, but Dr. Thompson's work in the lab, that was also CIRM funded, has demonstrated that a embryonic stem cell derived cell transplant in an animal model has mitigated the damage to the neurons through a variety of different potential mechanisms. And the really spectacular thing about it is this is a tough indication, really tough biology. But she and her team have amassed the scientific data that now have led to her being in what's called a pre-IND phase, getting all the information that we need to go to the FDA to then get permission to go into a clinical trial. And this is a CIRM-funded program. So she's or somebody from her group will be talking about that a little bit more in the scientific session tomorrow. So if you're able to, please attend. And another program that has recently been funded by CIRM is Dr. Brian Cummings' traumatic brain injury program. I don't know if you're all aware, but traumatic brain injury is a neurologic deterioration that happens after an unexpected trauma. Most of the cases are from motor vehicle accidents, and then the majority of others are from sports and recreational activities where it's an unexpected event that leads to damage. And Dr. Cummings has been researching this and has had scientific data that supports that a cell transplant could mitigate the neuronal damage and loss after traumatic brain injury, as well as tampen down the inflammatory milieu or the environment that leads to downstream damage, as well as potentially even provide the support in terms of new neurons. So he has a translational project to pursue this, look at the mechanism, and get it ready so that it can get to the next phase of development to do all the things that are necessary to get to where Dr. Thompson is in terms of getting it closer to the clinical trial. And then here's another program out of UC Irvine. It's Dr. Henry Claasen's. I'm sorry. This is, Dr. Henry Claasen's project, and this is Rosie, who is one of the early patients who was treated in this trial. It is the cell product here is a retinal progenitor cell that's injected into the eye, into the anterior chamber of the eye, into the vitreous. And the idea behind this is retinal, the cells, the neural cells that are the light sensing cells are lost in RP and retinitis pigmentosa. And the idea behind this therapy is that the cells themselves actually promote rescue of these light sensing cells. And so there's been some promising data from that that's still ongoing. And they have what's called an expedited pathway designation from the FDA, which is based on some early clinical data that will allow them to kind of have frequent conversations to try to expedite the progress of this trial. And Rosie reports on her own self-report that now she's able to see her daughter's faces where she wasn't able to before. And just little things. And she and her mom just has noticed that you can actually see things. She doesn't have perfect vision yet, but just an improvement in her vision has led to quality of life as well as some functional improvements. But again, this is why we do clinical trials, to make sure that this is reproducible. But it is also important that we're reminded by our patients why we're doing them and why is the urgency. So it's great that they are willing to come forward, and she's several years out now from that. And this is, I mentioned that there was a trial being conducted at UC Irvine, a company called Brainstorm Therapeutics. And Brainstorm is actually an Israeli company. They came to California because of the Alpha Clinics Network, because of the resources here, and because of partnering with CIRM. And because of that kind of ecosystem and that community that they felt would add to their ability to conduct that trial. Next slide. And it's not going to be much longer here, because I did promise it was going to be less than an hour. But one of the things I want to highlight is, because of this CIRM funding model, this acceleration model, this expertise we built, this ecosystem we built, the NIH, one of the largest funders worldwide, had asked us to partner with them and to use the CIRM systems to find and fund cell and gene therapy approaches to a bold goal. So they also put a stake in the ground. They said, we're going to cure sickle cell disease. Why not? 1940s, Linus Pauling already had written that there's a molecular basis for the disease. We know there's a single mutation in one of the amino acids, right? Why can't we cure the disease? Its time is now. And so we are partnered with them, and we have now funded one of the first programs under that MOU. Dr. Mark Walters, who's also part of the whole Alpha Clinics Network, he's at UCSF, Benioff. And the Alpha Clinics Network is involved with the whole Cure Sickle Cell Initiative, along with American Society for Hematology and all of the other stakeholders. So we're very excited about that, and stay tuned. And then I'll just end it by saying that we're not just targeting rare diseases, we're also targeting large populations. So one of our phase 3 trials, and phase 3 is the trial right before getting approval. So the trial that Evie's in is a phase 2. That's getting close to being approval with a small number. This is a trial that's in phase 3 that needs large numbers. but it's to target kidney failure. So Dr. Defoe knows, we're both transplant surgeons, that this is the lifeline for patients until they can either get a transplant or sometimes patients can't get transplants. It's the only way they can clear the toxins in their body. And often you run out of ways to access them. The best type of access you can get is a native, forming an AV fistula with native vessels, but they run out of them. And then synthetic ones and catheters have all their complications and infection. Through this technology, it's using stem cells to repopulate this biologic tube that once it's there and it gets engrafted, when you look at this, it looks like a normal blood vessel, but with better properties because it's rigid. And so this company is doing this, and what's validating about it is that Fresenius, which is one of the largest dialysis access global distributors, partnered with them as a commercialization partner. So again, just like with the other programs we talked about, CIRM de-risks the programs early on. Here's a slide. We de-risk it so that they can get early data and then they can get partnered up. So for $2.8 billion of CIRM funding, California funding, another $3.2 billion has come in because folks said, oh, this looks like looks really promising, and then they come in with investments, including industry, which, as you can see, each year, 40 million in 2015 and 2016, 153. As you can see, last year, 1.06 billion in industry partnerships, and 2 companies went public. So it's validated also by industry, the efforts that were made possible because of CIRM. And finally, this all is done because this is a collective effort. CIRM, Prop 71 wouldn't have happened without the community involvement. It was because of the patient advocates and other scientists banding together and saying, we need to pursue this. This continues. It's integrated into how we do things. We also interact with all of the other stakeholders, scientific foundations, National Academy of Sciences, to make sure we're doing this right. We have the Alpha Clinics Network. You'll recognize some of these folks. There's Dr. Bota there, and other members to deliver these to patients. We have our Industry Alliance program to continue to promote the probability of our programs making it out to commercialization and leveraging the initial investment. And then I'm going to stop here by just saying, be careful of the non-regulated work that's happening. There are sources out there for legitimate trials, but even clinicaltrials.gov, always confirm it with your trusted scientists and physicians, because we are still, while we're making progress in the field, there's unregulated activity that's occurring that's taking advantage of patients. So I'm just going to leave it at that because I do want to leave time for a discussion and questions. Thank you so much for your attention.
Wow, what a fantastic talk. Dr. Millan, thank you for an insightful journey through this exciting field of stem cell and regenerative medicine. I'm going to try to reach in here and
This looks really,
find something
this is
for you.
great.
There we go. I found it. There it is.
Oh, wow.
So here's a small token of our appreciation.
Oh, thank you
And
so
it
much.
says,
I was trying to grab it. I was trying to grab it from you.
UCI School of Medicine, Dean's Distinguished Lecturer, Maria T. Millan, MD, September 11th, 12th, 2019. Thank you so much.
Thank you
Very heavy.
so much. Oh, this is,
I think the good news is, first of all, what an exciting talk, what an exciting field. We certainly expect and hope that CIRM will get refunded and that all of these great advances that have really just started to hit takeoff speed can really take off. Hopefully you'll all join with the other California voters and vote the right way. So thank you.
so I wanted to say that although I can't be part of the campaign and speak either way, we do have our board members here and our chair of our board who can give an update from information we have for the citizens who are leading the charge in the campaign. So if there are any questions, but I'd like to just go ahead and turn it over to Jonathan Thomas for a few statements.
Yeah, let's have JT come on up if you would. And
Thank
then
you
we
very
can
much.
ask the audience if they have any questions after he says a few words.
Thank you very much, Maria. That was an excellent talk.
Thank you very much.
Thank you.
So go for it,
Oh, okay.
A-
I'll
a-
stay right here. There are a lot of places
A
to stand
fitting,
here.
a fitting inaugural talk for your series, which is going to be a smashing success. I can certainly tell. So thank you, everybody, for coming out to hear Maria tonight. Hope it was interesting. So the question I've been asked to talk about, and as Maria said, we're a state agency. We can't advocate. We can't electioneer. We can just state facts and hope that the facts are things that people are interested in. So in this instance, we had our $3 billion, and as it happens, there comes a time when even with a $3 billion, you run through it, giving out money for best-in-class projects, which is what we fund, and what all these outstanding projects are at UC Irvine. So we literally are going to be up against, in our next board meeting, which happens to be on Halloween, we will have officially more projects recommended for new research dollars than we have money to fund. So we knew this date was going to be coming, and the question is, how do you sustain CIRM and all this tremendous work that's been going on for the last 14 years? So the fellow who put the proposition on the ballot in 2004, Bob Klein, who was my predecessor as chairman of the CIRM board, is coming back to do a re-up measure on the theory that we have this tremendous portfolio of projects. We try to get as much of it pushed along so that there are interested partners out there, pharma, venture capitalists, the big biotech companies like Amgen, et cetera, to joint venture. But most of the stuff that we do hasn't hit that phase yet. So if we aren't successful in getting more funding, all this stuff's going to hit the wall that's not partnered up, and that, because CIRM is largely the only funding source of what they do, will be the end of all these great projects. So Bob, because of that, is determined to come up and have another measure on the ballot for the voters. It's going to be on the ballot in November of 2020, and it's going to be CIRM sort of updated to include developments that have happened since 2004. There have been a lot of interesting things in science that have been created since then. Gene editing is one you've all heard about as an example, so that you'd be able to fund stuff that wasn't even contemplated at the time of the original measure that exists now, as well as new stuff that's going to come down the pike, and nobody knows what that's going to be. There are going to be major new developments that further advance the ball that are not even on the drawing board yet. We want to be able to fund that too. So the measure is going to go in, and just very briefly, the technique, if you're a citizen and you want to get something qualified for the state ballot, it's daunting. There are two ways to do it. One, you can convince the legislature to put it on, but if you want to have something that you control, that you can dictate the terms and conditions of, you got to get it on yourself. And to do that, you need to draft a measure. It then goes to the state, and they review it and determine what they call title and summary, which is what the voters will see if and when it ever makes it to the ballot. Then, following a short public comment period, comes the big ticket item. You need to get signatures to qualify the measure for the ballot. And the amount of signatures you need to get is a function of how many people voted in the last statewide election, which in this case is 2018. And you'll recall, a lot of people voted. Without getting into one's politics, there are forces at play that are drawing people out to the ballot box these days, and 2018 was a big example of that. What that means for Bob is you need to get a percentage of that in signatures to qualify for the next thing, so the fact there was such a huge turnout means he needs a lot of signatures. And a lot means about 800,000. Bob is I don't know if anybody here has met Bob besides Oz, and I'm sure Leslie you have, and a few others, Marie of course. Bob is a sort of larger-than-life character, and he doesn't do anything small. So he needs 800,000. He's going to go out and get $1.5 million. That's his goal. And the reason for that is, if you're out there talking to people and say, hey, we got this stem cell measure coming, and this is what it's going to do, we really want your support, people hear about it, and then they sign their signature as part of this process, it invests them in it passing. So if you can get 1.5 million people, that's $1.5 million starting towards the amount you need to get your measure passed. That process takes 6 months, up to 6 months. If you get that, then the last step is the Secretary of State, for all measures that are attempting to get on the ballot, qualify them for the ballot as of June 2020. And then it's all about the race to the election, and you see what happens. How many of you guys have voted on school district general obligation bonds on your election ballots? I'm sure most everybody. Well, you know that those require 2/3 vote to pass because it's a tax on the voters, right? That's tough. 2/3 is a big, big number. So one of the interesting things about the state is you only need 50% plus 1 vote. And that 17% differential makes for a much higher pass rate at the state level, such that over history, state bond measures of any kind for schools or prisons or water or stem cells have passed 90% of the time. Doesn't matter how big they are, doesn't matter what the subject is, 90% of the time. So Bob is hoping that that holds true in this instance as well. So we'll know as of November of 20 what the likelihood is of this thing passing. Just as a non-participating, non-advocating member of the public, we believe the chances, as we sit here today, as they were in 2004 when it got 59% of the vote, are better than not that it will. And if it does, it will set CIRM up for another 20 plus years. And CIRM is the leader of the industry in the world. Nobody had the $3 billion to put out. So California is the big person in the game on this. And if that happens, the contribution that the wonderful scientists here at UCI and all throughout the state will make towards finding cures for all these terrible diseases that still loom will be significant. So stay tuned, watch what happens, and that's about all I can say on the subject. So thank you.
Thank you.
We still have a few minutes if we have any questions from the audience. Please wait for the microphone to appear if you don't mind.
Maria mentioned stem cells for brain injury. Does that include stroke?
For stroke?
Asked about the stem cell treatment for TBI. Did that include stroke?
The Dr. Cummings program is for TBI, but we have funded programs, clinical trials in stroke as well, and we actually have some of the investigators and scientists who've been involved in that. May I turn it over for you to just give a couple of comments on the stroke trial?
Sure. My name is Lenny Grossman. I'm involved in a couple of stroke trials, and a couple of them are completed at UCI, and some are still ongoing. So stroke, we do have a trial that essentially you can contact our stem cell clinic, and we will provide you the information. The easiest way to contact and get enrolled if somebody of your loved one had stroke is essentially to find the PISIS-3 website, or as I said, you can go through the stem. And essentially, with the previous trials, there were some successes. One of them was from STEMedica. Essentially, they showed that in the phase 2 trials, they were able to improve the patient condition after stroke. So the research is ongoing, and of course, UCI will be part of it. We are trying to kind of bring new trials to our facility, and just keep tuned. Does it answer your question? Or?
So there are no approved cell therapies for stroke currently, and it's a complicated and complex, condition, which I know it's hard to hear as complex. However, with all this information that's being gained from the other trials, by being able to collect the data and do this the right way, that is better than some of the things that are being tried where it's not being done under clinical trial. And so hopefully just by being able to analyze data that's collected the right way, it will point to approaches, whether it be cell therapy or others. But currently, we don't have a cell therapy that's close to being approved for stroke. It's what in that category of more to be done. Is there research being done on ataxia? There is research being done on ataxia, but there is none yet in clinical trials. So some of the kind of the early work in terms of the discovery and looking at the mechanisms and looking at the biology and the different signals, that is being done, but nothing that has yet gotten to where we have a candidate for it. Neurologic disorders, neurodegenerative disorders in general, are some of the toughest indications. So having programs that are this far along in terms of having scientific data to point to at least to a candidate where then you can test it in a way that you can measure whether it works or not is a key, whether it's a negative or positive data and all the data in between is a key step toward getting there.
We have time for one more question.
Could you give us an update on the generation of insulin-producing beta cells from stems?
Right. So there are a couple of advancements that have been occurring, and interestingly,
Maybe you could explain to the audience what he means.
okay, so Dr. Defoe asked what the progress has been in the diabetes world in terms of getting a cell therapy to treat diabetes, especially type 1 diabetes. The idea behind it is simple, right? The reason that folks have diabetes is they don't have functioning islet cells. The islet cells are the cells in the pancreas that produce insulin. Insulin is what regulates our blood sugar and is very important in metabolism. And as you know, diabetes, in addition to having problems with glucose control, have also downstream effects in terms of blood vessel issues, infections, and neurologic consequences. So the idea is if you are able to get a cell therapy, and it's not just exogenous, insulin, which imperfectly manages the glucose, but if you actually have a biologic cell that senses the glucose, secretes the insulin and other hormones to keep things in what's called homeostasis stable, that keeps us healthy, that's a better approach. We have funded a program out of San Diego called ViaCyte, which is a company that is using an embryonic stem cell derived product that gets matured to the point that they are then putting it in a thin wafer the size of a credit card. These cells are implanted in the body and then they mature into these islet cells, as well as glucagon, producing cells as well, the other side of, and that company is currently in the midst of a clinical trial, and we should start seeing some information from that soon. But there's nothing definitive yet that's been reported by that company. While all this information is happening, there are also other efforts in this area, including one out of Harvard, out of Doug Melton's lab. Our chair was just there visiting that group. That group actually had spun out a company called Sema Therapeutics, and that company was recently had a huge investment into it. And so although we don't see all the data sets because it needs to be done the right way, the fact that there's investor confidence in cell replacement therapy for diabetes, I think is a positive sign. Doesn't mean it's a done deal, but it's a positive sign. So nothing yet close to approval. I don't know of a pivotal trial yet with cell replacement, but it's getting there.
Mr. Thomas wants to say a couple last words, and then I'll thank everybody.
So I want to, we would be remiss here on an evening about CIRM to not pay tribute to one of your own who has been one of our most important members. Oz, would you stand up, please? You know, this isn't what he wants. So the CIRM board has been in place since 2004. I believe, Oz, there are only 4 remaining original members of that board. Actually, there are now only 3. And Oz is one of those. He was appointed. We have 10 spots on the board that are patient advocate spots, which are people who represent conditions and families of certain different types, although really what they do is represent patients writ large for all conditions. And they're an incredibly important voice because they bring what it's all about, that perspective to the board in discussions on what to fund. With Oz, we had a most unusual twofer because not only is he a patient advocate, but he is a distinguished neuroscientist here at UC Irvine. So he brings both the perspective of the patient, in his case, the spinal cord injury community, and his expertise as a scientist, which is most impressive. So the board, and through the board, the citizens of California have greatly benefited by having Oz as a member of our board, and UC Irvine should be very proud to have somebody who's had such a material impact on what we do. So one more round of applause for Oz. Thank you, Oz.
So one final thanks to all of you that came this evening to our inaugural Dean's Distinguished Lecture Series and helped make it a great success. Of course, Dr. Millan had something to do with that. Thank you again.
Thank you.
So please stay tuned as we'll be bringing you additional speakers from industry and academia who are making their marks on the world of healthcare. So thank you again for joining us. That's all. Thank you.